Yeah.
Good.
Dr. Chemerinsky, can you hear us?
Yes, we can hear you.
Perfect. We can hear you as well.
Okay, perfect. And you can see Dr. Adashi's slides?
Yes. Yep. And we can see him and you.
Perfect. Okay. Can I go ahead and introduce, or are we waiting for anything? No? Okay. So we have the distinct honor of hearing from Dr. Eli Adashi today. I'm a new faculty member, and this is my first opportunity to introduce the Grand Round speaker. But luckily, this is an easy one because Dr. Adashi's CV is 83 pages long. Dr. Adashi is a tenured professor with the Department of Medical Science of the Warren Alpert Medical School of Brown University. He is also the former Dean of Medical and Biological Sciences at Brown University. Dr. Adashi graduated from Tel Aviv University School of Medicine and went on to residency training in OBGYN at Tufts, followed by fellowship training in REI at the Johns Hopkins University School of Medicine and postdoctoral training in reproductive biology at UCSD. He also holds a degree in healthcare management from the Harvard School of Public Health. There's no way to do justice to his CV in this brief introduction, but I'll give you a small taste. He's a former president of multiple OBGYN and REI societies. He has served as examiner and director of the Division of Reproductive Endocrinology of ABOG. Dr. Adashi has served as a mentor to over 50 postdoctoral trainees and fellows and is the author of over 820 peer-reviewed publications and book chapters and reviews. Dr. Adashi was the recipient of continuous NIH funding from 1985 to 2005, inclusive of a Research Career Development Award, and it would take the better part of this hour to list all of his many awards and honors. Suffice it to say, he is an internationally recognized leader in REI. It's such a treat to hear from him today on the very exciting topic of in vitro gametogenesis, producing eggs and sperm from buccal smears. Please join me in warmly welcoming Dr. Adashi.
Thank you very much, Anat. And let me know if you need to change the volume in any way to hear me.
Sounds good to me.
Okay. Well, it's a pleasure to be back in Newark, even if via Zoom. I used to be there in person quite a bit over the years, going back to the tenure of Dr. Gerson Weiss. This is really my first Zoom lecture, if you will, at your institution. Maybe we can resume the in-person variety at some later date. Speaking of which, those of you who are interested can look up a paper of mine in the American Journal of Medicine that was published this year that deals with the pros and cons of the in-person versus the Zoom grand rounds. As you can tell, by dint of my old-fashioned inclinations, I probably come down in favor of the in-person grand rounds. At the same time, I acknowledge what I refer to here as the triumph of virtuality, which is the triumph of Zoom, of course. And today we are, of course, capitalizing on this very capability, which wouldn't have been possible pre-COVID. This is truly a novel development. What we will be speaking about today is the future. About 50 years or so after the birth of IVF, In 2028, we will be celebrating or recognizing the 50th anniversary of IVF. The leading but not sole father of which was Bob Edwards from the United Kingdom, who ended up receiving the Nobel Prize in 2010. The only reason his colleagues did not receive the Nobel Prize had really nothing to do with their level of contribution, but had simply to do with the fact that they were no longer alive. It so happens that the Nobel Committee only awards the prize to living persons. And so some of Professor Edwards' leading collaborators were never treated this honor. I might also add that Professor Edwards at the time was unwell enough so that he could not go to Stockholm. It was his wife who ended up traveling to Stockholm to receive the award on his behalf. Today we will speak about in vitro gametogenesis, and if I had to predict, I would say perhaps a future Nobel Prize as well. And it will be quite apparent who will be the recipients from some of our conversations today. But this is clearly a breakthrough that makes reality as we knew it for who knows how long completely different. And we will, of course, elaborate on all of that as we go. It is a little difficult to pinpoint the beginning of this story, but upon checking with the leading protagonist here, a certain Mitsunori Saito from Kyoto University in Japan, the first paper he identified for me as starting the story was published in Cell in 2009. For the longest of time, Professor Mitsunori Saito, who is shown on the left-hand side of this slide, collaborated closely with Katsuhiko Hayashi, who at the time that I met these individuals back in 2013 in Kyoto, Japan, those two were working together for a long time. At some point, Professor Hayashi established himself independently at Kyosho University, and these days he publishes his papers independently. But for the longest of time, these two were inseparable. And there is very, very, very little question but that they are the fathers of in vitro gametogenesis, and that very little competition exists to this day. It is notable, if not surprising, that there's very little work on IVG in the United States. By that I mean basic science work. And so as we speak, you will see that from the outset to the present time, these two gentlemen really deserve all the credit. We will mostly speak today about the rodent simply because that story is more or less complete. But we will, of course, mention the human with the caveat that the process of in vitro gametogenesis in the human has not yet been carried to completion, and as such is not ready for clinical application in any way, although I don't see any reason to exclude that from happening before too long. When we speak about pluripotent stem cells, which are at the heart of our story, we make a distinction between embryonic and induced varieties. And what we mean by that is that irrespective of the technology we use, we can create cells that in principle can go anywhere, can become anything. In a word, we could replace, for example, amacrine neurons in the retina in people who were born without it. We could potentially administer dopaminergic neurons into the striatum of individuals who are suffering from Parkinson's disease. And in yet another dreamlike scenario, we could potentially replace motor neurons in the spinal cord of individuals who experience paralysis. So the possibilities of pluripotent stem cells becoming any other cell type, including gametes, are really infinite. And in a way, what we will be speaking about today is a small subtype of what stem cells are all about, because they can effectively change human reality as we knew it for generations, now that we have replacement parts, if you will. Now, as advertised, we will speak today only about stem cell-derived gametes, but I thought it was important to emphasize the broad range of possibilities. Science magazine, which is a good barometer for what's hot, understood that IVG is making history. And it dedicated a full spread to it in 2021. The authors you recognize by now are really the main, if not sole, drivers of this field. The New England Journal of Medicine, not to be outdone, commissioned a similar paper for Mary Herbert and Azim Surani from the UK, titled Oocytes from Stem Cells, which dealt with the same issue and provided them with a counterpart. Last year, in Washington, D.C., I chaired a 2.5-day workshop of the National Academy of Medicine that dealt strictly with in vitro gametogenesis, which is to say that the National Academy of Medicine also recognizes, first of all, the importance of the subject, and secondly, the inevitability thereof, which is to say, it's coming. The only question is when. And so over 2.5 days, speakers from around the nation spoke about the multiple facets of IVG. Amongst the people who participated, in this case via Zoom, were, of course, our Japanese leaders of the field who contributed their effort. To give you yet another sense of the significance of IVG, I'm showing you here what amounts to the agenda of the Nuffield Council on Bioethics, which, as you may know, is the most significant bioethical institute in the UK. Every year, they publish this type of schema, which the human reproduction last year listed in vitro derived gametes at the top of their list, which is to say they too recognize how central this is to reproduction and what an important role it will play before too long. Yes, they deal with time limits on embryos in culture, womb replacements, preconception screening, whole genome sequencing of embryos, genome editing of gametes and embryos, and egg freezing, but at the top of the list, in vitro derived gametes. I don't think we'll have any difficulties agreeing that this is a disruptive technology. Time will, of course, tell, although I suspect the answer is favorable, whether this is achievable in the human. It's already been achieved in the rodent. And likewise, we will have to concede that the timeline is uncertain. As we launch into this story, it's good to remember the pioneering work of Aldous Huxley, who, in his famous book, Brave New World, in 1932, made several predictions, one of which was that eggs will be produced via ex vivo ovarian cultures. Well, this is not exactly in vitro gametogenesis, it's mighty close, which is to say that this gentleman was right on target, as he was when he predicted that embryos will be born via in vitro fertilization. I don't think any of us question this prediction. He also predicted, although that is yet to be accomplished, if in fact it should be accomplished, that newborns will be born via ex vivo ectogenesis. In a word, he could see ectogenesis for what it is, and it's still being pursued, that women will be freed up of the process of carrying the fetus and of the delivery thereof. Whether that's desirable or whether that's agreeable is a long debate and probably deserves a separate lecture. But that was one of the predictions made by Mr. Huxley. Needless to say, he was right on target when he predicted IVF. In chapter 1 of Brave New World, the director of hatcheries and conditioning says, one egg, one embryo, one adult, normality. Making 96 human beings grow where only one grew before, progress. In other words, they could see the writing on the wall even then. And today, there's little doubt that with IVG, the number of gametes that will be produced will be essentially unlimited. We don't need 96 eggs or sperm in a well, but if we chose to proceed this way, the technology will make it possible. Now, the very notion of IVG was facing, at the outset, early, I would say, theoretical obstacles, not practical obstacles. Because at the time that our Japanese friends began this effort, the so-called soma germline barrier hypothesis was reigning supreme. This was the brainchild of Professor Weissman, who lived in the 19th century primarily, and who, in his book, The Germplasm, A Theory of Heredity, maintained that, as was customary at the time, that the germline gives rise to the soma, which makes the germline immortal in a sense, and that the soma, for its part, never gives rise to the germline. In other words, you could never take a skin cell and make an egg or a sperm out of it, which is exactly what we're doing now. And he concluded that for these reasons, the soma was mortal because it couldn't be perpetuated. As I just said, in hindsight, this wasn't correct, but it was perfectly predictable at the time. What we will speak about next will be pluripotent stem cell formation technologies, because absent such, we can't go to the in vitro derived gametes. One such technology is known as somatic cell nuclear transfer, or SCNT for short, and it was produced or devised in 1958 by John Garden, then at Cambridge University, who published on the pages of Nature the possibility of creating stem cells, in this case using frogs. He essentially took the nuclei of somatic cells of the frogs, injected them into enucleated oocytes, and created, if you will, a hybrid zygote that developed into a blastocyst, gave an inner cell mass, from which one could derive embryonic stem cells. The second technology was developed in 2006. It's called the induction technology, and was contributed by Shinya Yamanaka from Kyoto University, whose findings were published in Cell. What Professor Yamanaka did is start with a somatic cell, transfect it with select transcription factors, and produce what was to be known as induced pluripotent stem cells, not embryonic, induced pluripotent stem cells. As you can imagine, both gentlemen received the Nobel Prize in 2012. And it is strictly due to this breakthrough that we can now proceed with in vitro gametogenesis, or for that matter, producing any cell type in the body that we care to produce, particularly where replacement is called for. In short, in vitro gametogenesis, or IVG, is all about taking a somatic cell, let's say a skin cell, converting it via one of these technologies to an embryonic or induced pluripotent stem cells, and then guiding that stem cell towards a gametogenic fate, which is the contribution of our Japanese colleagues. What are the prerequisites here? Well, you need to know what cues should be applied to the stem cells so that they become eggs or sperm. You need to know the sequence by which those factors are applied. You need to know the timing by which to apply these signals. And you need to take into account the so-called niche wherein the germ cells are embedded. I don't need to tell this audience that oocytes are surrounded by granulosa and theca cells, and that sperm are surrounded by Sertoli cells. And there is a crosstalk that's transpiring all along. And so understanding the niche and what it contributes to the development of a gamete is an essential knowledge in this process as well. It's a little difficult to nail the development of IVG, or the first paper, if you will. But in 2003, there was a paper in Science, senior authored by Hans Scholler, then a postdoc at University of Pennsylvania, now the director of a Max Planck Institute in Germany. As the title suggests, and as the figure on the right lower hand side confirms, something akin to an oocyte was produced. And that was done from mouse embryonic stem cell. That same year, a Japanese contribution maintained the same outcome on the pages of the Proceedings of the National Academy of Sciences. In 2004, on the pages of Nature, George Daly, who is now the dean of Harvard Medical School, also reported on the derivation of embryonic germ cells. And René Reopera, who at the time was at UCSF, reported the same. In the years that followed, several key papers in key journals were published. This is hardly an exhaustive listing, but it gives you a sense of what happened. This was in the rodent, and this was in the human. And so when we come to 2015 or thereabouts, we could say the germ cell specification pathway is not conserved between the rodent and the human, which has obviously significant practical applications. And the genetic networks involved are quite distinct as well. And so what we knew in 2015 is that we could take an embryonic stem cell or an induced pluripotent stem cells to a primordial germ cell-like cell. But we had no idea as to how to go from there to a haploid post-meiotic mature gamete, an egg or a cell or a sperm. We understood the extragonadal component. We did not understand its intragonadal counterpart. And so the final frontier was what I call in vitro gametogenesis all the way, which has now been accomplished in the rodent, but has yet to be accomplished in the human. On the female side, it was, when I say female side, I mean the rodent female side. It was Katsuhiko Hayashi who in 2016, on the pages of Nature, delineated the entire process for the generation of eggs. He started with stem cells. He combined them with somatic cells. He led them to become oocytes. M2 oocytes, which are shown on the left-hand side, which upon fertilization gave rise to a blastocyst and eventually pups. In a word, in the rodent, at least, in the female rodent, we can recapitulate in vitro gametogenesis from A to Z. On the male side, much of the same has been accomplished in the rodent. This work is the contribution of Mitinori Saito. And once again, we can say with conviction that in the mouse, at least, we can take a stem cell and create functional gametes at the end of the day. But the human paradigm was and remains lagging at this time. The furthest we have gone is illustrated here in a paper that was published in Science in 2018, senior authored by Mitinori Saito. He started with human primordial germ cell-like cells, combined them, for lack of a species-specific somatic cells, with xenogeneic somatic cells. Nevertheless, he was able to take those stem cells all the way to the oogonial stage, but never to the M2 stage. And so in a human, in vitro gametogenesis is still a goal that needs to be accomplished. And while one cannot predict anything with certainty, it seems all but inevitable that this feat will ultimately be accomplished by Professor Saito and or Katsuhiko Hayashi or both. I also predict, although again I have no control over the process, that a Nobel Prize will be awarded to these two gentlemen, but time will tell. I will not dwell on this except to say that much of the same is now true for the male side. We're not there yet, but we're trying in the human. So producing M2 oocytes and spermatozoa is still in the future in the human. Now, assuming for the moment that we have accomplished that feat, there will be some very clear scientific and translational benefits. For once, we will have for the first time an inexhaustible supply of germ cells. We were able to do what is known as null mutant analysis, that is to say, knock one gene out at a time and ask what happens, and thereby define the functionality of individual genes. We could also, for the first time, meaningfully study so-called germ cell somatic interactions, which we know must happen. But the reality is we don't know much about it. On the clinical side, we may be in a position for the first time to reverse honest-to-goodness germ cell failure. People with no gametes or in some way defective gametes that could potentially be cured. And that could be accomplished through autologous gamete replacement, if appropriate, or heterologous gamete replacement, if that's required. You could gene edit in cases where a genetic pathology is in place. And for the first time, and we will not discuss this in great detail today, it's a separate lecture of mine, we are finally on the verge of accomplishing same-sex parenthood. And if we ever meet again to speak about that, I will share with you where that science stands today. But in my mind, there is little question but that in a matter of decade, 2, 3, who knows, when the science progresses, same-sex partners will be able to be the biologic parents of the children. Needless to say, in the case of 2 males, there will be a surrogate required to carry the pregnancy. But other than that, we're talking about children who are the genetic offspring of 2 men or 2 women. And that's changing society as we knew it since it existed. It's a notion that is completely foreign to human history, but is about to be accomplished. For IVF, there will also be implications. IVF is not going to go away, but it will be modified in all likelihood. Instead of injecting women with gonadotropins with an eye towards stimulating the growth of multiple eggs, and then subjecting them to a, quote, minor surgical procedure, retrieving those eggs, we should be able to mature eggs in vitro using a somatic cell as starting material. Now, you would still need to transfer the embryo, but this will simplify IVF in ways we did not envision before. It will also do away with what we now routinely pursue, that is gamete donation, because one could produce gametes in alternative ways. We will not have to rely on donors, if you will. And to a large degree, this will convert IVF to a laboratory procedure, not entirely, but in a significant fashion. As I mentioned, irrespective of IVG, we will still need endometrial preparation and eventually embryo transfer, but that's really the tail end of IVF, and it diminishes the burden that women have to cope with today. Whether or not this will improve affordability and therefore access to IVF, which, as you know, is expensive and is not available on a universal basis, is always open to question. We have seen this promise before, and we have seen this promise fall by the wayside. Somehow we always find a way to jack up the prices. And so even though every logical fiber in our being is telling us that IVG should be more affordable than IVF, only time will tell. As I said, human ingenuity when it comes up to jacking up prices seems to be limitless. I don't have to tell this audience that something along these lines can't just, meander into the clinical setting and be applied. This will have to be subjected to rigorous investigation and regulation. And ultimately, it will have to come under FDA jurisdiction. specifically an application. I mean, clinical trials would have to be carried out, and the results will have to be submitted to the so-called Center for Biologics Evaluation and Research, which is presently headed by Peter Marks, MD, PhD, who will likely convene a committee of experts that will deliberate the merits of the application and will issue a verdict. This will fall under what Sieber calls HCTPs, or human cells, tissues, and products. They obviously deal with a lot of things, including, of course, drug approval, to name one. Which brings us to where we started, to the fact that IVG has now become part of the mix that we call reproductive endocrinology. And consequently, the ethics thereof has become also an important piece of evaluation by bioethical institutes. This is a remarkably promising future that is still unfolding. It's a little too early to celebrate, but many of us feel that now is the time to talk about it, lay the groundwork, introduce the basic principles to practicing OBGYNs and to reproductive endocrinologists, so that when and if this happens, there has been some groundwork laid down and some appreciation of the strength and weaknesses of this technology exists. Some have, I think, appropriately referred to this whole field as gametes to order, which is technically correct, and is really part of our brave new world that even Mr. Huxley was hard-pressed to fully appreciate, which wasn't his fault. But he simply lived too early to witness what I believe is a tour de force on the part of Japanese investigators who are making all of this possible. So stay tuned, and should there be any questions, of course, I'll do my best to address them. Thank you very much.
Thank you so much, Dr. Adachi. I don't see any questions in the chat yet, so we'll open it up to the audience that's in the lecture hall.
Thank you so much. Sorry, we were muted. Can you hear me, Dr. Adachi?
I can, thank you.
My name is Sarah Morelli. I am really grateful that you came and gave us another talk. Every time you speak, we are reminded of the true brave new world that we indeed live in. I'm not the director of hatcheries and conditioning, but I am a reproductive endocrinologist and really just thought this was fascinating. Although I'm sure that was not limited to the members of my division. I just had two questions for you. In the rodent studies in which the cycle has been proven, are there any studies beyond birth of pups or any follow-up of the pups and how they do?
Well, that's a good question. I haven't seen any published reports on that. I think it's a legitimate question. We clearly want to be sure that whatever gestation results, it is normal and comparable in every way. But I really haven't seen anything that specifically addresses this issue. I suppose that has to do with the intoxication we all felt by the very fact that we could accomplish a gestation. But your point's well taken. And I agree with you that this is, even in the rodent at this point, where it's doable, I think this is necessary and essential. And pre-implantation genetic analysis should also be feasible in the human, if not today, then very shortly, very soon, and ought to be carried out. So point well taken.
Thank you. I mean, I think we all agree that we do become sometimes easily and quickly intoxicated with, new developments. And, especially in our field, there are things like PGT that, move our pendulum so quickly. So, the point of regulation is taken. And the only other question I had to that end in terms of regulation is, or safety. I understand that there is a tumorigenesis risk with therapies that are derived from induced pluripotential stem cells in other tissues. I don't know if that risk applies in this circumstance. Do they talk about tumorigenesis risk in the literature, either in the rodent or the human?
Not in this context, but clearly, if I were sitting in that room with the committee that the FDA convened, I will ask the same two questions that you just asked, because their job is safety, to some degree efficacy. But I would say first and foremost, we have that structure in place for safety reasons. And issues of tumorigenesis, among others, clearly need to be brought up. And if it's not been studied in the trials that have been submitted for approval, the committee, is well within its rights to require that. And more likely than not, if the right kind of insight exists at the FDA proper, they can in fact request that before they ever convene a committee. In other words, they can review the application, they can deem it insufficient, they can return it to the authors and request that they carry out certain additional analysis before it ever gets to the committee. So there are clearly mechanisms in place that are designed to protect human welfare. And I definitely hope that this will transpire, which is why I mentioned it towards the end of my presentation.
Well, it's exciting. Thank you so much. I will see if there's anyone else in the room or on the chat that would like to ask a question. Oh, Dr. Chemerinsky, is there anyone on the chat who's asking a question?
No, there are no questions in the chat.
Do you mind just announcing the CME code,
Sure.
Dr.
Yeah,
Chemerinsky?
there's no questions in the group. Okay, perfect. The CME code is 25382. 25382.
Dr. Adachi, if there's no other questions, I want to thank you for coming to visit us virtually, and I hope we can return to the tradition of in-person in the future. But thank you for reminding us of the future every single time you speak.
Thank you. My pleasure.
Thank you.
And if you can, send me the recorded video. We make use of it.
Yes, absolutely. I'll send it to you.
Very good.
Thank you so much.
Thank you.